Hello and welcome back. Today we are going to talk about non-Mendelian genetics. And basically what that means is we're going to focus on types of inheritance that do not follow Mendel's three laws. Specifically, we're going to be looking at the law of dominance and how, when we were talking about Mendel, there was a specific dominant and a specific recessive allele. Well, in non-Mendelian genetics, that's just not how it happens. There's two specific types we're going to talk about, incomplete and codominance. We're going to talk about how they kind of go against what Mendel thought was happening. So let's just dive right in. The first pattern of inheritance we're going to talk about is incomplete dominance. And what this basically means is that neither allele is completely dominant over the other. Right, so again, going back to Mendel's law of dominance, not all genes have alleles that are only dominant and only recessive. So in this case, the way that we show the alleles is we use the same letter for both types of alleles. It's just one of them has a prime and one of them is just the uppercase letter, and that's it by itself. So let's look at this example below, right? We have big R, and that means red. And then we have R prime, we call that prime. It's just like an apostrophe, but we call it prime. And that's white. So those are our two main alleles, red and white. But when we have the heterozygous genotype, we actually get a blending, that's a key word there, blending of the two of them together, right? Red and white blended together makes pink. And incomplete dominance shows an incomplete, like, masking of the other allele. So instead of being just red or just white, we have this new, brand new third phenotype, which is a blend of the other two. So let's show this in an example of a Punnett square. So I want to cross a pink snapdragon with another pink snapdragon. Now, if you don't know what a snapdragon is, it's a flower. So we're going to cross a pink flower with another pink flower. So to start off, remember our steps to the Punnett square, we need to start with our parent cross. And so, using the allele key from the previous slide, right, RR is red, R prime equals white, and our parental genotype is RR prime, because that's the heterozygous genotype, crossed with RR prime. Now, once we have this, we're going to just throw these genotypes, these parental genotypes, up onto our Punnett square. So let's do that right now. And then as we go through, we're just going to fill in the Punnett square how we normally fill it in, right? We're going to go across and go down, and we're going to fill in these squares where these alleles would intersect with each other. So in this top box, we have RR. In the next one, we have RR prime. Then we have R prime again, and then R prime. Now the key here, the only real difference here is understanding what our phenotypes are, right? So our next step is to do the genotype ratio, and that's easy. You're just counting how many of each genotype you have. And in this case, I have one RR, right? That's in my first box. I have two R primes. And we can see that in the offspring. And then I have one R prime. Now for our fifth and final step here, we are going to have our phenotype ratio. And the difference here is that there are three possible phenotypes. So you do have to notate all of the phenotypes. So in this case, I have one red snapdragon, right? RR notates red. I have two pink snapdragons, right? RR prime is pink. And then I have one white snapdragon because I only have one offspring that's going to be R prime. Okay, so that is incomplete dominance, where you get a blend of the two alleles to make a third brand new phenotype. It's a blend. That's the key. Blend. Okay, let's talk about our other type of non-Mendelian inheritance, and this is called codominance. And codominance, you can probably guess just from the name, is that they are both dominant. Both alleles show dominance. And what that means is that the hybrid, or the heterozygous genotype, shows both of the phenotypes equally. So instead of blending into a third brand new, never before seen phenotype, instead we get a phenotype where I see both colors equally. In this case, the notation is a little bit weird and it takes a little bit of time to write, but you do have to superscript the letters. So it's the same letter that you start with, but then the superscript letter is different depending on what the allele is. So let's look at our example below, right? We have CB, and that, we're going to talk about chickens here, by the way. So CB is black feathers, CW is white feathers, okay? So our C's are all the same, always the same, but the letter that you're superscripting is notating what the color of the feathers actually is. So CB, the B stands for black, CW, the W stands for white, and then CB, that heterozygous mashup, you're going to have black and white feathers equally shown. So it'd be like spotted versus blended, okay? So let's practice this again with our Punnett square. Okay, so for this Punnett square, we're going to cross a white feather chicken with a black and white feather chicken. And in this case, we're going to do the same thing that we did with our incomplete dominant Punnett square. We're just going to do another practice one. So our first step is to bring over the allele key that was on the previous slide. Okay, so I have CB for black feathers and CW for white feathers. Now I'm crossing a white feathered chicken, so I already know that to be white feathered, my genotype is going to be CW. And I'm crossing that with a black and white feathered chicken, which means the heterozygous genotype. So pulling up my parental genotypes, I see that CW crossed with CB. That makes sense. I know that. I knew that because I was able to remember my allele key, and I was able to remember that black and white, because I'm showing both, that means heterozygous. So now I'm going to throw that up into my Punnett square. Easy peasy. And then we bring our alleles across, we bring them down, and we fill in our Punnett square like we've been doing this whole time. Okay, so in this case, my CBs go across, my CWs go across, and because my white chicken only has CWs to give, we're going to bring only CWs down into all of the genotypes. Now what's interesting about this, and you didn't see it in the incomplete dominance just because that was the example I gave, but in this one I do want to point out that even though I don't see any CB, right, I don't see any black feathered chicken in my offspring, I do have to notate that there are 0 of them. So I do have to actually be clear that there are 0. So let's talk about this genotype ratio. As I mentioned, you do have to notate all of the possible genotypes and how many are present in the offspring. In this case, I have 0 CB. So I don't have any in any capacity, right? No parent with CB and no offspring were, but 0. I have to say 0. If I don't put 0, I'm gonna mark it wrong. And then I have 2 CBCWs, right? I have 2 black and white chickens. And then I have 2 CWCWs, right? I have 2 white feathered chickens. I can see that in the offspring. Now for my phenotype ratio, again, I do have to notate if there are 0 in the offspring. So for this, I have 0 black feathered chickens. I know it seems redundant, but we're going to practice good genetics here, so please notate 0. And then I have 2 black and white feathered chickens, and then I have 2 white feathered chickens. So again, I'm just taking those genotypes, translating them into the phenotypes they represent, and I'm writing that down, okay? So please remember that you do have to give me 0 if there are 0, okay? Great job. We're going to practice this in class, so if it was a little bit confusing, don't worry. I've got your back. I'm really proud of you. I'll see you soon.